maybe all this global pandemic stuff has changed.
Yeah. So let's start the webinar and let's just cut this video.
You are. Hello and good evening to everyone. On behalf of the Office of Global Engagement, I'm very excited to welcome you to the 12th webinar from the IRIS webinar series. My name is Richu George Philip and I'm a part of the Office of Global Engagement. From the AI and Data Sciences Technology Cluster, the research initiative being presented today is Integrative Biology and Systems Medicine, or IBSC, led by Professor Himanshu Sinha. IBSC is poised to capitalize on data to develop novel solutions by applying ML and AI to improve India's healthcare. Dr. Himanshu Sinha is a population geneticist interested in how the information flow from genotype to phenotype is dynamically modulated by genetics, environment, and evolution. He works with yeast populations to develop transferable knowledge to understand complex human diseases. He extends this knowledge to study complex relationships in clinical and genomics data sets to build models to define better the clinical outcomes for improving public health. As an IBSC coordinator, he has been instrumental in positioning it as a leading center for computational and systems biology research in India. Our moderator for today is Dr. Jason Pitt. Dr. Pitt is a computational biologist interested in how environment, lifestyle, and genetics mediate oncogenic cellular transformation. He focuses on scalable genomic data science, emphasizing the development of platforms that enable efficient processing and querying of large cancer genomics datasets and applying these platforms to understand cancer better etiology. Also joining us on the panel today is Dr. Shinchini Bhatnagar and Dr. T. Ramchandran from Translational Health Science and Technology Institute, and Professor Karthik Raman, Professor Manikandan Narayanan, Professor Neerav Bhatt, and Professor Balraman Ravindran from IIT Madras. A warm welcome and thank you to everyone on the panel today. A note to the participants, please use the Q&A to enter your questions and upvote questions that interest you so that the moderator can prioritize them later. Over to you, Professor Himanshu.
Thank you, Richu, for the introduction. So welcome to this IRIS webinar for Center for Integrative Biology and Systems Medicine, IBSC. It's a center for applying data science and AI to improve healthcare in India. And I'm Himanshu Sinha, PI of the center. Yeah, oops, sorry, why did it move? Give me one. Sorry, yeah. So before we begin, I just want to define how a disease is defined and its treatment is determined. So if you consider any disease, let's say, for example, breast cancer, which is one of the very common disease across the world, it all depends on the information that is available at the time when this disease is detected. For example, in early 19th century, the only treatment for breast cancer was surgery. Later on, in the late 20th century, after decades of research, we were able to identify several risk factors that induce breast cancer and develop diagnostic tools and therapeutics like tamoxifen and Herceptin. Now, in this century, early on, we sequenced the human genome and several novel insights were developed, which led to development of several targeted drugs, inhibitors, which were used as treatments. And with the availability of large cancer data sets which identified gene networks, we were able to identify several combination therapies, which now become as the standard care. And as you can see, facilitated by molecular understanding of mutations, genes, networks, and pathways, we have a large number of treatments and targets which are very, very specific for each mutation. So as you can see, the definition of disease and its treatment is very much dependent on the understanding of the whole system. And this comes to a very ancient statement by Aristotle centuries ago, where he said the whole is more than the sum of its parts. And that's what is the essence of integrative biology, which means that research using experimental and computational quantitative technologies to characterize biological systems, be it diseases, at molecular, cellular, tissue, and population levels. And this is facilitated by quantitative understanding of how properties at each level determine the behavior of the whole system, and that is the whole complex system, and that's what integrative biology allows you to do. So what is systems medicine? Now, if you consider the breast cancer example, we saw that there were several molecules and several targets. Essentially, for any treatment of any disease or even a phenotype, what it is dependent on is a multi-dimensional layers of information that we have to analyze. Now, these layers include the environmental signals, which could be the environment the individual is living in, the diet, which then interacts with the genome of that individual, which dynamically changes the cell as well as the molecular pathways that get activated, which results in gene, which is this transcriptome, and proteome expression changes, leading to changes at the tissue level as well as the population level. And nowadays, we also know the microbiome, the gut, adds another layer of complexity to the readouts that we have. So a systems medicine approach will help integrate this multi-layer biological data, which includes DNA, RNA, proteins, molecular pathways, cells, organs, and individuals to investigate how complex changes in a disease happen. And this would help us achieve a better understanding of the disease, leading to a more personalized treatment for that particular disease. So the question comes, where does IBSC stand? What is IBSC? So IBSC, as I'll show you in a few slides later, it's an interdisciplinary group which is dedicated to developing innovative approaches and algorithms that integrate multi-dimensional data, as I was just talking about, DNA, RNA, proteome, and environment, across scales. Scales would be different levels, like at molecular, cellular, and individual level, to understand, predict, and hopefully manipulate these complex biological systems. And being at IIT Madras has a huge advantage for IBSC. First, for example, we have a large interdisciplinary faculty. What we can also do, we can apply well-established engineering principles onto biological systems. The manpower we have, the human resource we have at IIT Madras has a strong quantitative aptitude, which is very useful in developing these novel approaches and algorithms. And we are very much integrated into the Robert Bosch Center for Data Science and AI. The IRIS webinar, which you heard, one of the IRIS webinar which you heard just before this one was from that center. And we have a National Cancer Tissue Bank here at IIT Madras, with which we collaborate very strongly. So what I'm going to do today is to give you a very brief introduction of the 4 research projects in which IBSC involved in. And they would be very, very brief introductions just to give you a flavor of what kind of research we do. First is a project on preterm birth risk modeling. Now, as you might know, preterm birth is a birth before 37 weeks of gestation. An average human gestational period is for 40 weeks. In India, actually 3.6 million babies are born preterm, of which 50% die soon after birth. So there's a huge burden on childhood mortality. To better understand the causes of this preterm birth, high preterm birth in India, the Department of Biotechnology, Government of India funded a Garbhini preterm cohort run by Translational Health Sciences Technology Institute, Faridabad, to study 8,000 pregnant women in Gurugram district in Haryana. These women were enrolled early in the first trimester and observed throughout the pregnancy, collecting several clinical, obstetric, ultrasonographic, anthropometric, and socioeconomic parameters. And till date, we have collected about 10 million data points. And this data set is the data set we plan to use to develop a dynamic preterm prediction model to help stratify women for timely clinical interventions to mitigate preterm birth risk. Now, gestation age determination is absolutely critical for estimating preterm birth because it's a time-dependent phenotype. It's not a clinical feature where you have pathology or some other feature that defines preterm birth. However, unfortunately in India, we use a US population-based gestational model. We therefore have a less accurate determination of gestation age due to population level differences between the US and India. So to determine the gestational age of Indian fetuses, we developed India's first models for both first trimester, Garbhini GA1 model, and second and third trimester model called Garbhini GA2 model. And the first model we recently published in April 2021, and the second model is almost done, and will be soon publishing that. One of the advantages of these models is that it helps us make more precise estimations of gestation age, resulting in more accurate preterm birth estimates. And some preliminary data shows that the high risk that we see of preterm birth might not be as high as stated because we were maybe underestimating the age of several Indian fetuses. So that's the first project. The second project I would like to describe is about the genomics of Indian people. Now, as you might know, India is very unlike any country in the world. It consists of around 4,000 unique subpopulations, which can be divided based on geographical, ethnic, and linguistic groups, which mostly follow endogamy within their groups. A 2017 study showed that the genetic variation in the Indian population, which is shown here on this PCA plot, is far higher than the European, American Caucasian population or the Chinese population, which in turn means that if a disease marker is identified in an American European population, that may not be causal in Indian populations, and we might have our own set of disease markers, and that can be very deterministic for diagnosis of a complex disease like cancers. So keeping that in mind, again, Department of Biotechnology, Government of India has recently started Genome India 10K project, which is to do a whole genome sequence of 10,000 healthy Indians spanning these geographical, ethnic, and linguistic groups. And what we hope to achieve at the end of this project, which is an ongoing project, is to identify India-specific disease variants, which will not only help develop a database for India-specific genetic diseases and their causes, but it would also kickstart a population-specific personalized medicine in India, which is the requirement, given that if you see the kind of diversity we have, some populations might respond differently than other populations within India. We're not even talking internationally different populations. The third project, what I would call is a true example of integrative biology. What we know that tissue-tissue and cell-cell communication are fundamental for development of any phenotype in a complex organism like a human. However, what happens in most most studies are either very tissue specific. They will only study the brain, or they'll only study the digestive system or the heart, without understanding how these diseases progress across tissues and organisms. So using computational approaches, which when applied to gene expression data from each of these individual tissues, we plan to dissect both within and cross-tissue networks to understand how these networks rewire when diseases occur. And one of the first diseases we are trying to understand this for are neurodegenerative diseases. And the final fourth project is very contemporary. Just to introduce what is metagenomics, it's the study of the genetic material from a mixed community of organisms isolated from environment. Now, MetaSub is an international project, first started in New York, in the subway of New York, where the environmental metagenome, that means what is the collection of organisms in the environment in mass transit systems, is what is that? The question is, what is the collection of these genomes in these mass transit systems? So a project like MetaSub is a very useful tool for public health officials to enable smart and responsive cities, because now you can actually get a signature of what is present in the environment even before that becomes a pandemic or an epidemic or a localized problem. So in MetaSub Chennai, what we plan to do is to sample Chennai Metro for bacteria and viruses, including COVID-19. So this would allow us to understand what is the environment like in different parts of the Chennai Metro system. And that's something that we now will start now that the Chennai Metro has started running again. So coming to the team, as I said, it's an interdisciplinary team, which you can see 3 faculty are from Department of Biotechnology with expertise in population genetics, computational and systems biology and modeling. And we have 2 faculty members from the Computer Science and Engineering Department with expertise in networks, data science, and AI. We have also assembled a panel of eminent scientists and industry experts to provide sound research advice through their critical insights in their domains and providing a solid direction to the center. And we are hoping that their inputs would drive the research in the center to achieve greater heights. Now, coming to the societal impact, it's pretty obvious that we have a very firm footing in the healthcare domain. Garbini and Genome India projects are 2 national missions or projects of the government of India, and we are very deeply involved in it. Our research outcomes of Garbini project we are very sure will improve child and maternal health, thereby help India achieve the WHO sustainable goals of reducing childhood and maternal mortality rates by 2025. That's the goal. And one of the project I did not describe today is another project, which is an Indo-EU project called Incentive. And we are involved in developing adult influenza vaccination programs. And this now takes a new impetus due to COVID-19 pandemic. Finally, just to give you a flavor of the kind of research that has been done at the center in past few months, and that got a lot of popular press traction. Here's a report in Hindu describing a study about genome scale modeling of metabolism of gut microbiomes, which we now know are one of the primary determinants of diseases in humans. This is another news coverage of metagenomics of gut microbes, again in Hindu newspaper. This is the coverage for the development of the first trimester dating model, which is first in India, and it was covered both as a review article as well as a news item. And finally, this is one of the most recent coverages, actually still happening, like still flashing around the news, about a paper which describes a model to determine cancer mutation signatures. So as you can see, a lot of press interest is there because of the implications that some of our research projects are on public healthcare. So there are several ways to engage with IBSC. You can collaborate on research projects, help conduct training programs. We have 2 fellowships, one postdoctoral fellowship and one postbaccalaureate fellowships for students to come and work with us. And finally, we are seeking in young international faculty where at IBSC, they can actually work with unique Indian healthcare data, which is not available anywhere else in the world. And these are some of the upcoming events. We plan to have this joint EBI IBSC workshop on biomodeling in December 2021. The last one was a huge success. This was before COVID. We have another international symposium slotted for February 2022, which we plan to do on a broad topic of clinical genomics. The last one was in 2020. That was just before the COVID lockdown around the world, and that was sponsored by EMBO and was very well attended. And we, throughout the year, plan to host leading international and national scientists by organizing seminars and colloquia. So finally, I'll end by saying thanks to you all, the audience, all our students who work very hard, the faculty collaborators that we have, and the funding agencies for supporting us. Thank you.
Okay. Well, thank you very much, Himanshu, for that great overview of IBSE. So given that I'm moderating this session, I was hoping I could throw one question out at you right away. I think it's very admirable that the center is focusing on multiple projects that are very specific and very relevant to the Indian population. Interactions with the global community and the global profile of the research that you're doing here in India. Where are some possible collaborations that you see?
Right now, I think one of the first things that one of the issues with many of the Indian projects, especially with healthcare, is there are several governmental restrictions because these are all federally funded projects. But what we plan to do, like for example, with NUS, we do plan to have collaborations on how to develop novel methods for say, for example, analyzing genomic data sets that come out of India. And I'm sure with preterm birth, we do want to collaborate with the South Asian countries, which are sort of genetically, we would assume to be more closely related to India and therefore have similar kind of patterns. And that's the other thing. There are several, like, as I said, I didn't mention in detail, but incentive is a strong, it's already an international project where vaccination programs in India will be done in conjunction with vaccination programs in Europe. And that would be another sort of large collaborations that we hope we'll be able to carry forward.
Okay, great. Well, why don't we take some questions from the Q&A if the panel doesn't mind. I think one here that's kind of jumping out at me that maybe has a little bit more to do with the field than just the center, but it'd be interesting to hear if IBSE has any plans of addressing this. But Anita asks, is there any plan to explore healthcare and AI from the perspective of analyzing healthy individuals as opposed to taking a disease-centric view to try to use those healthy individuals to try to understand what it might be like to live a healthier life?
Yeah, that I think is very good. So there are projects being done in several countries, like especially in US, where people are doing these 1 million healthy individual genome projects. The Genome India 10K project right now is only on healthy individuals. So we will get a lot of data about how healthy individuals, what are the genomes of healthy individuals. But yeah, you're right in saying that while most of the research is done to identify disease and disease markers, because the funding is easier first, and secondly, there's a direct impact that you can make on the disease. Studying healthiness is sort of relative, right? A healthy individual today may not be healthy individual 5 years later, right? So defining healthy individual becomes a challenge. But yeah, it's a very good question, and I think more and more work should be done to identify what are the reasons for being healthy, especially longevity, rather than healthiness at like middle age. In one say. I don't know if Karthik wants to add something to it. He can.
Yeah, I think that's a very important point that Himanshu made about the status of person changing through the process, and I think we are also finding some of these things during the sampling for Genome India, right? So some person may not be diabetic, but maybe pre-diabetic and so on. And yeah, I think as we accumulate more data, we're just starting off now, and I think we'll have some more answers to all these questions.
Yeah, and I think related to the India, or related to the data collection process, another question was asking, given the population of India itself, do we think that sampling 10K individuals will be enough, or where do you see things going from there? I mean, it probably is a little too early to say, but maybe if you guys want to give your thoughts on the sample size and potentially the future plans of collection of genomic data in India.
So I'll quickly answer that question and pass it over to Himanshu, who will give you the longer answer. But I think we should be calling this phase 0 of the project, right? So it's a very, very exploratory phase. We're calling it phase 1, and there's an obvious understanding that there are going to be bigger and larger phases that follow up. But this is a start to really see what is the scale at which we can operate and get the pipelines in motion and so on. And there is definitely a plan to, and this is already quite an expensive project. And I think we need to really iron out all the bugs and then go in at a much larger scale. Himanshu.
Yeah, so I think that's the whole idea. Like, this is one of the first times that India is doing such a large project. You're right, Jason, that this is really an underrepresentation. So even if I take a rough estimate of 4,500 subpopulations, I'm only sampling 2 individuals per population, right? Essentially, and that's already 9,000 individuals, right? So the sample size is definitely an understatement. But what I would say is that this is far more going to be a very rich resource, given the fact that in the 1,000 genome project that is very commonly used by researchers around the world, there are only about 10 Indian genomes. And we know Indian genomes are very unique compared to the Caucasian population. So I think this would give us a much better representation at the international level, where we will be able to define. And obviously, as right now, these are whole genome projects, maybe later on, more targeted exome sequences can be done, where we can now start determining more deeper level of variations that we are interested in, and especially looking at diseases that are prevalent in India, rather than trying to use data sets which are developed for a disease which might not be that prevalent in India, or might not be that consequential in India. So yeah.
Yeah, and if I can add on that, I think it's important to recognize kind of that old adage of, how do you eat an elephant? One bite at a time. And I think you have to start somewhere. With a population like this. And you mentioned 1,000 genomes, and when that started, that was a giant collection of data. That was only 1,000 individuals. And you look at what Singapore has been doing recently with the Singapore 10K project, where even their 10K project, they only had 5,000 samples to start with, but there was a rich amount of information that came from that. And now we're scaling up to do about 100,000. So I think it ends up being that snowball effect as well. I think it's great that you guys are able to even start with 10,000 and just see what you can find from there, which might fuel the need or just the desire to have more genome sequences.
Yeah, and also it sort of establishes the sort of the framework on which we can actually build, because 10,000 can easily be amplified to 100,000, and that can, once it's established, so it allows a framework.
So I'm seeing a couple of questions here related to kind of being able to collaborate. I know you mentioned a couple of the post-bac fellowship and the postdoctoral fellowship as well, but I guess in terms of just anyone who might want to get involved with IBSE, what should they do? What are some opportunities there? Whether they're a clinician, whether they're a researcher within India, whether they're an international researcher, maybe an international student.
So we have they're both IBSE specific programs. Like, as I mentioned, postdocs, we can hire any postdoc. We don't have any limitation whether they are from India or not. We can have postbacs. Right now, most of the things are online, so it's a lot easier anyway. But we do have several other avenues through institute, and I think Karthik can add to that. where at the institute can facilitate these sort of collaborations or these trainings or these recruitments through an institute-wide program where they can then come and work specifically with IBSE. Karthik.
Yeah, I mean, there is just various programs for people at any level. They could be a faculty in a different institution or a student from abroad and so on. There are programs for everyone. I mean, they just need to reach out to us. We can really give them inputs. But we have international PhD programs. We have international internships. We have national, of course, PhD programs, internships, and fellowships. So we basically have a lot of scope for collaboration with the center, and we have funding also for our own students to visit other labs and initiate collaborations and things like that, both within the country and abroad. So there's a lot of opportunity for mobility which can be exploited to nucleate collaborations.
So I'm seeing a lot of people interested in this question as well, and it's obviously quite a broad one, so feel free to answer how you both would like, but whether it's specific to IBSE or just general, the bioinformatics field in general, of where do you really see the role of AI and data science playing, really aiding bioinformatics, of maybe what specifically could be applied, or what types of data would we want to use, what type of approaches? I mean, what's your guys' vision for really bringing AI to bioinformatics?
Ravi, you want to go ahead?
Sure. Let me take a stab at it. Right. So first of all, I don't know any bioinformatics or anything, and I work very closely with the people in IBSE because I bring the AI to the table, and the domain information comes from people who understand it. Right. So what is really amazing about, Karthik has his favorite quote from Kunut. Right. So the next decade or next century of problems for computer science is going to come from biology. Right. I might be mangling the quote. Karthik, you want to fill in the exact quote?
Yeah. So biology has 500 years of exciting problems. It's at that level.
So that's the quote, right? So there are so many exciting opportunities when we start looking at whether it is the systems biology side of things or the bioinformatics side of things. There's such a wealth of data. We are talking about 10,000 individuals being small, right? So, but you can imagine the amount of data that's going to be generated from 10,000 individuals, right? That's going to run into like several petabytes of data for the time being, right? And so for a computer science person, for somebody who works with data and AI, right? I typically salivate at these prospects. Right, there's so much that we can do. So for some very specific examples, right? There is so much work that has been done on looking at many of these biological phenomena as graphs, like as entities and interactions between them. And there is, and that's actually driven a lot of research in AI recently on looking at how these kinds of graph structured data can be processed and what kind of insights that you can draw from that. Right. And so a lot of our collaboration actually revolves around that and advances. In fact, we even push the boundary, not just on applying AI to biology, we even push the boundary on the actual AI techniques itself, so that we can make them successful in the biological domain. And people might have heard of AlphaFold, which was a very successful AI program on solving protein folding. Right. So there are so many avenues in which the scale of problems is so large when you come to biology that classical algorithms kind of flounder. Right? So you need to look at data-driven computation like AI methods. So I can keep going, but the marriage is pretty obvious for me.
I guess if I can just add on, I share a lot of the same views. I think one of the biggest mistakes I've seen biologists make bioinformaticians make over the past 10 years, and I often joke about this, is that if we think we're special, and that, in the sense of there are some things that are very specific to biology that we're trying to solve. But if you look at the world and you look at the effect that data science and AI has had and the methods that have been applied in industry to all of these different problems, there are so many ways that you can look at a problem and see how it is very similar to a problem that we're looking at in biology. And you can start looking at ways that we can take methods that have already been used in other disciplines, even just to start, and start applying them in biology and then see what happens. And then, as was said, then we can start pushing our own boundaries and developing new methods once we see how they start performing. But I think really, we can benefit from looking out into the world, looking into tech, looking into statistics, physics, thinking of what we can do based on what has already been done there. No.
Yeah, so just to quote one of our faculty members, like Professor Raghu, he has this very nice slide where he shows I think a chemical processing plant. I think it's a petroleum processing plant. And he says, if anything goes wrong in this, there's going to be a disaster. This is exactly what human body is like. It's a system. If anything goes wrong in the system, which is very balanced, very optimized, you get a disease. So if you understand the processes and the optimizations and the algorithms that apply onto this factory, you can directly copy paste onto biological systems and get new insights. And that's exactly one of the things that IBSC is very much interested in, because that's the advantage of being at IIT, that we can start applying these very, very fundamental, very polished, very well established engineering techniques and algorithms onto biological data and get new insights. And that's a huge advantage.
Yeah. Well, and just to add on of how that view permeates internationally as well. I recently hired a data scientist who worked for Rolls-Royce, who his job was to look through aircraft log reports to predict when an engine would need to go for maintenance. And I pointed out to him, this is the same as a clinical problem when you're trying to predict when a patient should receive a drug. It's just different input data. But it's exactly the same. I told him, I want you, you're useful, you know these techniques, come, let's come and do some work together. So I think it's very, very, very true that.
And on the same lines, we have mechanical engineers, undergrad in mechanical engineering, working on preterm birth risk predictions, because they are essentially predicting failures, right? So
exactly. Okay. Well, I think we've covered most of the main topics here.
So Jason, this is Mani here, if I may.
Yes, hi Mani. Yeah.
In a comment, yeah. Hi, Himanshu. I enjoyed your presentation. So one thing that I think the participants here may really benefit from is an example of how you can design a population-specific medicine in an Indian context, so something that is already out there. And the subpart to that would be, if you could discuss, is how can a larger citizen base or a larger researcher community participate in figuring out those population-specific medicines themselves, right? Like what data resources would IBSC provide eventually that they can themselves do the analysis in their own labs or in their own homes, even if that is possible. So to contribute to discovery of these population-specific medicines, right? So an example of that and how they can engage.
Oh, you're asking me?
Yes. So, I mean, it's kind of like, yeah, if you would kind of explain it, it will be even better than me giving an example. Yeah.
So for example, I can, like one of the most successful projects that is going on right now is preterm birth prediction. So we have had several clinicians come up with their ideas and their data sets, saying that we have this very rich data set. Like, for example, there are hospitals that do these radiology, right? They are doing these ultrasounds. And now that we have a model, they really want to see whether their prediction of the due date for delivery, is it accurate or is it not? So these are the kinds. So I think what I'm trying to say is that we are very open to collaborating with hospitals, clinicians, not only just for preterm birth. We had a incipient collaboration with a major hospital here on image analysis of PET scans, right? That's another area where image processing is a huge field where data science and actually deep learning kind of AI can be applied to understand or even categorize images for normal and diseases. So there is a lot of scope. We are open ears to everybody. If you have an interesting question or you have an interesting data set which you have analyzed a bit but really want to see and deeply understand what does that data mean to you, we are open for discussions, and let's talk. I think the first step is communication, and one thing I would say, one of the advantages at IBSC is that because we are sort of, as I mentioned, and like there are two computer scientists whom we have, I think, talked enough biology, so they understand biology very well, hopefully. And we are biology from biotechnology department, so the communication can happen, right? One of the biggest hurdles for a clinician to talk to a computer science or data science guy is data science seems to be too sort of distant. And I think by approaching
Can we start this webinar?
Yeah. Yes, Professor. Shivram, you can just stop sharing the screen so that we can just get it started.